day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled ADC Development, Early On and Off Target Testing to Fail Fast. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. At this point, I'd like to thank Discovery Life Sciences who developed the content for this presentation. Discovery Life Sciences is a global leader in biospecimens and specialty lab services, enabling the discovery and development of groundbreaking therapeutics and diagnostics. With a robust portfolio of human biospecimens and cellular starting material, preclinical, ADME tox solutions, and cutting-edge specialty lab services, they provide their partners with the tools for data and expertise they need to succeed. From genomics and proteomics to cell biology and molecular pathology, their comprehensive services offerings support every phase of the scientific journey from research through clinical trials. Now it's my pleasure to welcome our speakers, and you'll notice there we have them all on screen. And first I'd like to welcome Dr. Emer Clark. She's the Vice President of Cell Biology, Discovery Life Sciences. So there she is. She has over 25 years of experience in stem cell biology, spanning both clinical and industrial settings. She plays a pivotal role in driving innovations and managing Discovery's scientific operations, including research and development and client services. Dr. Clark is also actively involved in client affairs, offering one-on-one consultations, study design, and data analysis. And next, I'd like to welcome Dr. Sean Fall. He's a Vice President of Lab Operations, Cell Services, and R&D Biospecimens with Discovery Life Sciences. So there he is, Dr. Fall. He earned his PhD in microbiology from the University of Virginia and completed a postdoctoral fellowship at Fox Chase Cancer Center. With expertise in transcriptional regulation of lymphocyte development, Dr. Fall joined Discovery in 2017 to pioneer analytical pipelines for single-cell evaluation of primary human biospecimens. So now that we have the introductions out of the way, I'd like to pass the mic over to Emer. So Emer, when you're ready, you may begin.
Thank you very much, Sonia, for the introduction. And with my colleague, Sean Fall, we are actually going to present to you some information on antibody-drug conjugates, both looking at on-target and off-target effects. If we start at the right-hand side of this slide, we're going to first think about traditional chemotherapy. And in olden times, basically chemotherapy aimed at killing the cancer cells, but there was a lot of collateral damage, and an awful lot of healthy cells were also killed. And if you were aware of people that took chemotherapy a number of years ago, you may have noticed that hair loss would be one of the side effects. This is because hair is one of those rapidly dividing cell populations. Interestingly, the hematopoietic cells are also a very rapidly dividing cell population, which is why different types of chemotherapies have an effect on blood cell counts. And we'll revisit this in a little while. As things continued, there was some ideas that if you could get some immune cells to go and attack the cancer cells, you could have a better outcome. You hopefully wouldn't kill as many healthy cells. However, a lot of these immunotherapies were non-specific. And when the immune cells got activated, there could be side effects like the release of cytokines or interferons. So although cancer cells were targeted and killed, there was still some collateral damage with the healthy cell. More recently, there's been a construct known as an antibody-drug conjugate, and this is what our talk is going to focus on today. This is an interesting molecule. It has an antibody section, it has a chemical linker, and it has cytotoxic drugs. And the aim of this construct is for the antibody to drive the system to bring this construct to the cancer cell, and once attached to the cancer cell, to release these cytotoxic drugs. And in doing so, selectively kill a cancer cell, but not the normal cells. Now in this diagram here, you can see the antibody section. The linker is a chemical structure, and the cytotoxic drugs are chemotherapeutic agents, sometimes also called payloads. In this particular model, you will see that there are 4 cytotoxic drugs, and that's what they call a DAR4, with DAR standing for drug antibody ratio. You can have DAR2 ADCs, or you could have DAR 8 ADCs. And as you can imagine, the more cytotoxic drugs that are linked to the antibody, the more potent that potential ADC may be. So the first generation antibody-drug conjugates were either mouse or chimeric antibodies. The linkers, these chemical structures, were unstable, and sometimes there were low potency payloads or chemotherapeutic agents. By the time the second generation of ADCs arrived, the antibodies were humanized. There was more stable or cleavable or non-cleavable linkers, and the payloads had a higher potency, which hoped that they would kill more of the cancer cells. Now we're on the third generation antibody-drug conjugates. These are fully humanized antibodies, or FABs. They're circulating stable linkers, and there can be low potency or novel payloads that are now being used. Again, a small shift away perhaps from the chemotherapeutic agents of old and onto kind of payloads that now would be immunomodulators. So a whole metamorphosis of this constructs have developed even in such a short time. So there's unprecedented interest in antibody-drug conjugates, and 15 have been approved by the FDA already. What's interesting is there's lower success rates in the phase 1 and phase 2. But if you manage to get your ADC to a phase 3 clinical trial, there can be great success, and hence some of the interest in the market. Overall, ADCs have an approval rate of 9%, which actually is higher in general than the 3.4% in general oncology therapeutics. But because of the phase 1 and phase 2, people are kind of thinking, if we're not going to get to phase 3, we would prefer to fail fast. So let's ask the question, what would make a good ADC? You definitely want high stability. You want sufficient toxicity, low immunogenicity, and good target expression. If your cancer cell is exceptionally rare and the target expression is exceptionally low, the ability of the antibody to find it may be limited. And so why do we have so many failures? Well, one of the key agents or reasons is off-target toxicity, where parts of that construct are falling apart and they themselves are having an impact on the off-target toxicity. Another challenge may be the target is expressed on healthy cells. And a very good example of this is acute myeloid leukemia, where one of the main targets is CD33. But CD33 is also on myeloid cells that are required to generate neutrophils. So if the target expression is on both a healthy and a disease cell, there are additional challenges. One of the reasons for so many failures is really the premature drug release, which means the separation of these cytotoxic agents from the antibody. When separated from the antibody, they're free to float around the body and cause havoc. And there's one other potential challenge is that the antibody binds to sites it should not bind to, and you have off-target binding, and then of course when the payloads are released, they're actually being released to cell populations that they should not be released to. And at this stage, I'm going to pass over to my colleague Sean Fall, who's going to tell you some of the effects of on-target efficacy with antibody-drug conjugates.
Fantastic, thanks, Emer. So what I'm going to talk about in the next section is on-target effects. And what we're looking at here is a survey of about the top 20 ADC targets that are currently in clinical development. This ranges anywhere from preclinical all the way to approved listings. And if you look at this list, it's going to be a mix of both solid tumor targets as well as some hematopoietic targets. So for this part of the talk, I'm focusing mostly on our solid tumor targets. As you look at this list, there are going to be a lot of markers that are being targeted via other methods. HER2 is highly listed on here, PD-L1, as well as some of those hematopoietic markers like BCMA. But really what we wanted to be able to do is really address the ADC market in two avenues. One is having a robust biorepository to screen expression levels for both these known targets as well as novel new targets, and then in vitro systems to test for on-target toxicity. So we looked at this robust biobank through two different types of tissue matrices. So the first one is fixed tissue. So any tissue that is resected in the clinic is going to be fixed and embedded in FFPE. This is by far the most prevalent status for solid tissue. And so we have been able to acquire a lot of fixed tissue into our bank. This is looking at about the top 15 or 20 indications that we have. And we have this in two different forms. So informed consent blocks are new blocks. These were patients that gave us consent to have their tissue. And so they are a little bit newer. They've had a little more of the more up-to-date annotation on them. The larger proportion, however, are known as waiver of consent blocks. So depending on the country of origin, there is a standard amount of time that a block has to be retained at the clinical site to be reviewed later if the patient comes back or if there's any difficulties. However, after that fixed time, in the US it's 10 years, those blocks are available for research use. And so a lot of the blocks that we've acquired are these older blocks that are waiver of consent. However, even though these blocks are older, they do still have a lot of benefits. They can be requalified up to the most recent standards. They can provide a breadth of patient demographics that aren't necessarily available. And so just looking at the numbers that we're displaying here, we have a couple 100,000 blocks that have already undergone re-secondary QC through our own pathologists, and we have a rather large bunker of millions of blocks that we can survey as new projects are brought forward. And so as an example, this happens to be PD-L1 staining. So some of our blocks were old enough such that they didn't get PD-L1 staining on them when they were originally diagnosed or came from areas of the world where that wasn't standard of care. And so we've done a rather large, robust screening effort looking for PD-L1 expression. In this case, this is showing lung cancer blocks. And we've used this to get both combined positive score and tumor proportionate scores on a number of our blocks from relevant indications that would receive checkpoint inhibitor therapy. So fixed tissue is very, very valuable. There's a lot of it out there that we can screen. But we also wanted to have a bank where we could get really focused on what cell types within the tumor are expressing potential ADC targets, as well as being able to use that material to generate in vitro models. And so what we have started to do is take fresh resection tissue dissociated to single cells and cryopreserve this. And so what this essentially provides us is a bank of almost fresh tissue that can be used for a lot of assay types that require single cell suspensions, flow cytometry, in vitro cell culture, single cell genomics. So this is again looking at that same 15 to 20 indications, and in this case we have several hundred unique cases in the biobank that we can survey for receptor expression or initiate in vitro cell culture from. And so what we started to do for this bank is started to do a lot of correlation studies. So again, focusing on PD-L1, we're looking at 2 lung cases as well as a renal cancer cell case. These had already been qualified using IHC stains on matching FFPE blocks. And then we used our dissociated viably cryopreserved cells to check the PD-L1 expression by flow. And we saw a very good correlation where if it scored positive by IHC, we had a clear separation of a PD-L1 signal away from the isotype where they virtually overlaid each other if no PD-L1 expression was present. And so again, looking at the indications that more commonly receive checkpoint inhibitor therapy, we now can qualify these samples on their PD-L1 expression. But the nice thing about having these dissociated tissues is now we can look very specifically at tumor cells or immune cells or stromal cells and the unique subtypes within those to actually understand where else potential targets might be expressed. So now that we've got a bank that we can know the expression dynamics of an ADC target, we next wanted to move into a system where we could test these in vitro. And so we've been working for a number of years on various short-term culture systems, but more recently we moved to 3D long-term model systems. So these are the most physiologically relevant models that we can do for solid tissues. They are 3-dimensional. You can see one growing on the right side of the screen. And they do replicate most of the features of the original tumor. These are scalable, so we can do large-scale expansion of these, which I'll show in the next slide, and they're stable, maintaining the inherent genomic and transcriptomic profiles of the parent tissue. And what we set up is basically the idea that we can grow up these large cultures and cryopreserve them back as well. And so now we essentially have a replenishment system to have these models be used essentially for years to come. So the way that these models are established is that cryopreserved dissociated tissue with single cells is first placed in an embedded or static culture, which you'll see on the far left of the screen. These are your more traditional matrigel, Geltrex cultures, where you have the semi-solid matrix that the cells are embedded into and that they'll start to grow out of. At a certain point, once the culture has established, we will move these embedded cultures into what we call a suspension culture phase. So in this case, there is still a small proportion of basement membrane extract in the culture, but it's much lower. And these cells at this point will grow almost like a pure hematopoietic cancer suspension cell line would. So you'll get a lot of clustering, and you can actually scale these up quite high to high cell numbers because of this. So one of our biggest focuses when we were establishing these cultures was to make sure that the suspension culture that we get at the very end still represents the parent tissue that it came from. And so what we're going to show on the right side of the screen is whole exome data that we did from the initial cryopreserved tissue through the embedded stage, the suspension stage, and then from that, a working bank that we have that we can actually use for testing. So what you're going to see here is basically the VAF of the viable conserved cryopreserved tissue is on the y-axis, whereas the VAF from each of these banks is on the x. And we saw very high correlation between all of these various allele frequencies from each bank as we progressed from the primary tissue up to the working bank. And then as we continue to check these VAF frequencies against each other, that they remain very, very high through these cultures. So we're maintaining that inherent genomic integrity of the parent tissue. And we've also shown this at the transcript level, although I'm not showing this for this talk. Importantly, they do still remain unique from each patient. So what you're seeing here is two patients. There's the 3309 patient on the y-axis, the 1104 patient on the x, looking at either its primary dissociated tissue or the tumoroid that was generated from that. And essentially there is no correlation for the VAF frequencies when we look at two unique cancer tissues or tumoroid cultures from each other. So these are unique going forward. So coming back to ADCs, we had all this RNA-seq data that we've generated off the tumoroid models, and we also performed unbiased mass spec analysis as well. And so this is looking at that list of 20 targets and basically whittling it down to those factors that are most relevant for solid tissues. What we saw was, first off, very good correlation between the relative expression we saw by RNA and by protein, and that most of these targets were expressed in both of these lines. So we were really focused, however, on the one that wasn't, which is ROR1. So if we look at both RNA and protein, it is present in the 3309 tumoroid, but not present in our 1104 tumoroid. And so because of that dichotomy, we want to confirm that. So using flow cytometry, we did show that the 3309 is indeed positive for ROR1 expression, while it is negative in 1104. But we also got very interested in the fact that HER2 was expressed with both of these as well. And so we did confirm, again, by flow cytometry that HER2 is expressed by both of our tumoroid models. And this allowed us to transition into some in vitro culture systems to try to test some of the HER2 ADCs that are present on the market. So to this end, we primarily started using cell titer glow 3D models. These are nice models you can grow up very quickly and get a very robust dose response curve in response to various compounds. And so this is looking at some of our initial trial work using more standard of care chemotherapy, such as 5-fluorouracil or etoposide, as well as a small molecule inhibitor against WEE1, and shown that we got differential responses between the 3309 and the 1104 tumoroids. However, when we look specifically at two different HER2 ADCs, especially two with two different payloads, we started to see differential responses between these two tumoroid lines, where the 1104 showed some slight sensitivity, but not really to either compound, whereas the 3309 showed a distinct response to one, but not to the other two, which is very interesting in that these molecules actually differ in their affinity for HER2. So this gives us a very robust system to screen lots of different antibodies, lots of different payloads to understand which ones provide the best on-target toxicity. However, while all of this works on target, we also are very conscious of the off-target toxicity, which I'll have Emer describe in the next section.
Thank you, Sean. So again, off-target toxicity is one of the rate-limiting factors for the use of the antibody-drug conjugates, and hematopoietic cells are very susceptible to off-target toxicity because they are continuously proliferating. The reason they're continuously proliferating is they have to supply the blood system with red blood cells, white blood cells, and platelets. And for example, a white blood cell may have a half-life of 9 hours. So if you do anything to disrupt the production of those white blood cells, there's going to be a significant reduction in the blood called neutropenia. A reduction of the red blood cells is called severe anemia. And if your platelets are affected, that's known as thrombocytopenia. So the way we actually measure off-target toxicity is we take the antibody-drug conjugate and we take bone marrow cells. We incubate them together in a liquid culture for 72 hours. And this technique was actually developed when we were looking at very, very large bispecific molecules, where we wanted to ensure that in a liquid culture system, the antibody would bind to both cell populations of interest. After 72 hours, we transfer the cells into a semi-solid matrix called methylcellulose, or megacult if we're looking at megakaryocytes. And we incubate these for 14 days at 37 degrees. And after this time, we take out the plates and we literally count colonies. And looking down the microscope, we can see in this particular culture, we're looking at an erythroid colony known as a BFUE, or a myeloid colony known as a CFUGM. The BFUE, the colony at the top, has kind of an orange-reddish tinge, and that's because that colony is composed of mature red blood cells. In contrast, the myeloid colony, or CFUGM, is composed of mature white blood cells. But each one of those colonies is derived from a primitive precursor. So what we're going to do now is we're going to look at the three components of the antibody-drug conjugate and say, where could the toxicity be coming from? Now, there are many, many chemotherapeutic agents, also known as payloads, that are being used in these antibody-drug conjugates, and they have different effects. So MMAE causes anemia, a reduction of the red blood cells, neutropenia, a reduction of the white blood cells, and peripheral neuropathy. DM1 is associated with thrombocytopenia, which would be a reduction in the platelets, and hepatic toxicity, whereas MMAF and DM4 cause severe ocular toxicity. So this graph shows the effect of those payloads on the erythroid, BFUE, or the myeloid, CFUGM progenitors. And they're divided by the topoisomerized payloads or the MTI payloads. Now, if you take a bird's eye view of this, what you can see is, in general, the same colored lines are in the same order, regardless of whether you're looking at the erythroid or myeloid series. And this is an absolute hallmark of off-target toxicity. This toxicity is not lineage specific. We're not taking out the red blood cells and sparing the white blood cells. It's just an off-target effect. And if you look at the lower section with the MTI payloads, you'll see that there's more than a 2-log difference in the off-target toxicity. And if you look carefully, you can see that MMAF, brown line, is way less potent than, for example, MMAE. However, ocular toxicity is one of those things that is exceptionally difficult for patients, and so basically isn't really tolerated. So now if we look at the ADC as a whole, this was an interesting project and collaboration with Zymeworks. They had developed a lot of ADCs, and the project we decided to do first is to take ADCs where the antibody, PALI, and the payload, 886, were consistent. And the only thing that changed in these antibody-drug conjugates was the linker. So in this assay, we took the entire ADCs, placed them in liquid for 72 hours, transferred them to the methyl cellulose, grew the colonies and counted them. And here you can see that there's an interesting spread of more than a log between the IC50s between them, where only the linker is affected. So let's talk about the linker. The first generation of linkers were unstable, non-cleavable, and acid-liable. The second generation had improved stability, but they were cleavable or non-cleavable and typically monovalent. And the third generation were more stable in circulation. I always think ADCs remind me of the motorbike with the sidecar. The motorbike, of course, is your antibody. It's targeting something, it's going somewhere. And the sidecar is your chemotherapeutic agent. And what's holding them together is this chemical structure called a linker. Now, if the linker breaks, as you can imagine in this situation, the sidecar has no engine and it wobbles freely wherever it will go. The motorbike can still go to the cell of choice, it still is driven, but really the chemotherapeutic agent is floating around the body and potentially causing havoc. And so a very nice publication came out in 2017 by Zhao et al. And they suggested that neutrophils caused their own demise. They suggested that the neutrophils, by releasing enzymes, caused linker cleavage, thereby allowing the payload to be released from the antibody. And the assay involves the generation of mature neutrophils from CD34 primitive cells with media and cytokine changes over a 17-day period. The compounds are added to this liquid-based, flow-based assay on day 11, when neutrophil generation has already started. And the readout of this assay is by flow cytometry, and it actually is a readout of neutrophils. And on the right-hand side, you will see 3 different ADCs with linkers A, B, and C, and this is real data generated from true ADCs with differing linkers. And we can see that the IC50 values between the linkers is quite distinct. And so sometimes when the target is chosen, when the chemotherapeutic agent is chosen, it's then important to evaluate which linker may be most appropriate for the moving forward towards clinical trial. So here are the data generated in this linker stability assay. Again, the monoclonal antibody is consistent, the chemotherapeutic agent is consistent, and the only thing changing is the linker. And there can be quite a divergence in the IC50 values between them. So again, let's just recap. What is going to make a good ADCs is high stability, sufficient toxicity, low immunogenicity, and good target expression. And you'll fail if there's off-target toxicity, and again, our focus is on the hematopoietic cells, but it can be on the liver cells or the eye, or if target expression is on healthy cells, which becomes an additional challenge in the development of the ADCs, premature drug release, the sidecar being released from the antibody, or off-target binding. And with those thoughts, I'm going to send it back to Sean to take us home with some additional thoughts.
Awesome, thanks, Emer. So what Emer and I have been talking about a lot during this webinar has been the early stages, the preclinical stages of ADC development, such that we can try to reduce the amount of failures in phase 1 or phase 2, particularly for off-target effects. But another thing that we think about quite a lot at Discovery is actually on the other end of the spectrum as we've gotten through clinical development. And one of my favorite thought experiments to do is how many therapies have failed not because they weren't legitimate, because they didn't work, but because they didn't work on the population of patients we tested. And so really this idea that we've got companion diagnostics being developed at the same time as the therapy really can make sure that we're targeting patients for the most therapeutic benefit. And so we spend a lot of time thinking about this. A lot of times we're using FFPE as our basis for this. Again, that's the matrix that almost all solid tissue tumors will be in. And so we've looked at this for several different technologies, either from singleplex to multiplex IHC, RNA-ish, or FISH, and really matching those technologies to platforms that are on the market, highly vetted, highly validated, and then getting those to a certain level needed for that study. And this could be as something as simple or as not as needed from a regulatory perspective as RUO, all the way up through IVDR, CAP/CLIA testing. And really this just makes sure that our patients are being treated that can have the best responses to these ADCs. And so two examples I love to show of this, the first being PD-L1. So this was a companion diagnostic that was generated, and this was some of the studies that were put forth in designing that companion diagnostic. And so on the left side of the screen, we're looking at 3 cell lines which had known expression patterns of PD-L1, either no expression, medium expression, or high expression, with their relative IHC stains below. So almost the inverse of how we did the PD-L1 profiling on the viably cryopreserved dissociated tissue. And so this really allowed us to see with known positive and negative controls that the IHC staining was working, and then translating that to actual real world tissues, which is what you're seeing on the right side. And this has really been fundamental for treatment on checkpoint inhibitor therapies. Patients on the indications that are on those are getting tested for PD-L1 to know if it would be advantageous for them to receive that therapy. The other is HER2, and so this is showing some HER2 staining examples on the left side of the screen. But the thing I really like about the HER2 example is that not only was the companion diagnostic established, but then training for the post-approval reading of this was also established. So that's what's shown on the right here, where pathologists were actually given on-site training in terms of how to interpret HER2 staining results. Really with something like IHC staining, being able to make sure that from pathologist to pathologist you're getting the same readout is highly important. So really having these trainings set up to make sure that we're getting the same reading regardless of individual is very, very important as we move forward. And so really all of this, what we've discussed today, has been this idea that if we can use in vitro assays such that we are finding problematic ADCs, be that the antibody, the linker, or the payload earlier, we can really raise those phase 1, phase 2 failures and eliminate them at a much earlier stage. This allows us then to move things faster through the clinic and get to those phase 3 where we do have a much higher rate of success. And really the idea that one of the things we like to say discovery is patients are waiting. The idea that there are patients out there that can benefit from these. So as much as we can do to shorten those early developmental stages and really focus on good candidates faster will be important as we move forward. So I'd like to thank everybody for joining the webinar today. We do have some questions, and I will now pass it back to Sonia.
Okay, well, thank you very much, Sean and Emer for that great presentation. I hope everyone enjoyed it. Now, I would like to invite our audience to continue sending in their questions using the comment section of the Q&A for the Q&A portion of this webinar. And if you are attending by a GoToWebinar, remember, please use the questions window. And for those of you watching on LinkedIn Live events, feel free to post your questions in the comment section. Okay, so now that we have that out of the way, I did receive a bunch of questions while you were speaking. So let's start with this first one here. This audience member is asking, you only discuss colorectal tumoroid models. Have you developed or plan to develop models from additional indications? It's a great question. Who would like to take that one?
Yes, I can take that one. So we use colorectal here. It's a great model system. A lot of the tumoroids that we're developing are based on that indication, but we do have others. We have lung, endometrial, and breast tumoroids that are in culture right now, and we're working on developing a handful of additional indications. So yes, it is outside of just CRC, even though we use CRC as an example for this talk.
Okay, thank you very much for that. I'm just here looking at some of the questions coming in. Let's go to the next one. Are you developing any other assays for evaluation of ADC function in the tumoroid models? Who'd like to take that question?
I can grab that one again. And then Emer, I might have your thoughts as well, because I know you do a lot of assays that we don't. But yeah, so the little animation that we showed is actually one of our INCU sites, and we are moving to using those real-time imaging platforms for ADCs. The one thing with the cell titer glow, it is a bulk readout, and so this way we can actually look a little more specifically into readouts against ADCs, specifically using that real-time imaging model. And then Emer, I didn't know if you had any other thoughts there.
Sure. So what I showed today was really off-target toxicity, but we can show on-target toxicity with leukemias. So for leukemias like acute myeloid leukemia and multiple myeloma, we have the ability to grow those cancer colonies in the metal cellulose with slightly different cytokines. And then we can ask the question, could the ADC have an effect on those tumor cells? What's interesting is with our system, we can really only look at floating diseases, whereas with Sean's system and the organoids, he can look at the solid organ. So we're kind of covering both angles between the two of us. And we're developing more assays for more of the leukemia-based ADC development.
Thank you very much for that. Let's go to this next question here. They're saying, thank you very much. I wanted to ask, most often is the cleavage of the linkers via the release of elastase through net formation?
The answer to that is we have no way to test that. It's the hypothesis and it's a very good one, but we have no way to actually test that in our in vitro systems. So all we really do is if we have antibodies that are consistent, payloads that are consistent, and linkers that change, we can evaluate the effect of the linker on neutrophil demise. And that really is what the assay looks at.
Okay, thank you very much for that. Okay. They have an addition to that question. They're also asking, I also wanted to ask, how are you ensured that heterogeneity of the antigen is being represented using the biobank and assays, especially in diseases such as TNBC, where we know patients of color are less likely to be willing to partake in research or sample donation? It's a great question.
That is a fantastic question. Yeah, I can take that one. This is something that we talk about quite a bit. And so when we are looking to acquire new samples for the biobank, one of the aspects that we take into account is demographics. And so whether we're getting waiver of consent, where we're talking to hospitals that have blocks that are older, that's actively what we ask. We also have the informed consent material that we get. We actually have our own employees a lot of times at these sites that talk to patients themselves to ask or answer any questions they might have. So it is something that we actively do look into and work on and are trying in as many ways as we can to make sure that the bank is as representative of the entire human population as possible.
There you go. Emer, do you have anything to add to that before we go to the next question?
Sean covered that beautifully.
Okay, all right. Okay, so this looks like it may be our last question, unless we have any questions coming in right now from our audience. So let's go to this one here. This person is asking, have you explored culturing any other cell types from tumor microenvironment?
Yeah, so I can start with this one, and then Emer, I'd love to hear your thoughts on the liquid tumor side. But on the solid tumor side, yes. So we have established fibroblasts from our tumoroid lines. So we have a matched cancer-associated fibroblast, as well as we've got PBMCs and other material. And so we are actually starting co-culture systems with multiple of those components together. So this allows us to profile a receptor expression on each of those, but also see if there are specific off-target effects within the microenvironment itself. And then Emer, I'd love to hear your thoughts on the hematopoietic side.
Sure. So, and the microenvironment in the hematopoietic system is extremely important, and it's often described as mesenchymal cells. And in many ways, these mesenchymal cells are like the soil in which the hematopoietic cells, the flower seeds, reside. You need both to work. It's often believed that the mesenchymal cells perhaps keep the stem cells in a more primitive state and therefore allowing them to develop when needed as required by stress in the environment. There are a couple of very nice publications out there looking at the growth of AML in the presence of a stromal cell line and also of multiple myeloma primary cells in the presence of a microenvironmental cell and perhaps a mesenchymal cell. And it is believed these would facilitate the extensive growth of these cell populations where in our own limited liquid culture systems, those cells mightn't survive for a long enough time to do appropriate analysis. So these models are being developed. They're not robust yet, I would say. They're not off the shelf yet. But I think they'll get there. We just need a little bit more practice, a little bit more time, and to understand what the microenvironment cell is offering to the cancer cell. And I think that's really the way we need to think about diseases, not as the cell in isolation, but the cell as part of the body and part of the whole hematopoietic system or solid organ system.
Fantastic. Thank you so much. Sean, do you have any other closing remarks before we close out the Q&A? Because I believe that's all the questions for now.
None from my end.
Okay, well, thank you so much, Sean and Emer, for that great presentation again, and thank you very much for answering all those questions. We have reached the end of the Q&A portion of the webinar.